making a new YouTube account, and I'm basically trying to study for a biology test, and I'm having difficulty understanding the text. And I just thought if I read it out loud, it could make some sense, and I thought maybe you guys would like to hear some of it. So here goes. If you're having trouble with biology, like some people, you can watch it. This is in Modern Biology by Holt, Reinhardt, and Winston, and it's chapter 10.1. I hope this helps. Nucleic acids and protein synthesis. DNA. What enables cells to have different forms and to perform different functions? Ultimately, the genetic source of this amazing diversity is deoxyribonic nucleic acid, DNA. I apologize if I completely butchered that. The primary function of DNA in organisms is to store and transmit the genetic information that tells cells which proteins to make and when to make them. Proteins in turn form the structural units of cells and help control chemical processes within the cells. Structure of DNA. Recall from Chapter 3 that the nucleic acid, DNA, is an organic compound. DNA is made up of repeating subunits called nucleotides. Each DNA molecule consists of two long chains of nucleotides. A DNA nucleotide has three parts: a sugar molecule called deoxyribose, a phosphate group which consists of phosphorus P, atoms surrounded by oxygen O, atoms, and a molecule that is referred to as nitrogen-containing base because it contains nitrogen N atom. Three parts of DNA nucleotides are illustrated in Figure 110. The deoxyribose sugar and the phosphate groups are identical in all DNA nucleotides. However, in nitrogen-containing base may be any one of the four different kinds. The four nitrogen-containing bases found in DNA nucleotides are adenine, guanine, cytosine, and thymine. It is customary to represent nucleotides by the abbreviations for their nitrogen-containing bases. A nucleotide containing adenine is represented by A. Likewise, C is cytosine, G is guanine, and T is thymine. So, you can kind of see right there. This is deoxyribose, phosphate groups, and nitrogen-containing bases. Figure 10.2 shows the structure of these four nucleotides. Notice that adenine and guanine have two rings of carbon, C, and nitrogen N atoms. In contrast, cytosine and thymine have only one ring of carbon and nitrogen atoms. Bases that have two rings of carbon and nitrogen atoms, such as adenine and guanine, are called purines. So, two rings of carbon are purines, and pyrimidines are bases that have one ring of carbon and nitrogen atoms, such as cytosine and thymine. Purines have two rings, pyrimidines have one ring. Obviously, I'm not going to pronounce these right. The double helix. In 1953, James Watson and Francis Crick suggested a model for the structure of DNA. The model proposed that the DNA is composed of nucleotide chains that wrap around each other to form a double spiral. Similar to a spiral staircase, the shape is called a double helix. The double helix structure of DNA is illustrated in Figure 10.3. I'm sure everyone's familiar with the double helix, but yeah. Watson and Crick relied heavily on scientific evidence reported by other scientists to construct the model. They cheated. The model was inspired in part of X-ray photographs of DNA crystals, like the one shown in figure 10.4. Now this is the famous X-ray that gave Watson and Crick the idea for their double helix, which they stole, but I won't get into that. Yeah. That has been studied by Rosalind Franklin and Maurice Wilkins. In addition, the model provided an explanation for how copies of DNA could be made and how genetic information might be stored and used within cells. In 1962, Watson Crick and Maurice Wilkins received the Nobel Prize for Medicine for their work on DNA. Rosalind Franklin had died in 1958 and thus was not recognized. And they stole her work, but I'm not going to get into that. Note in figure 10.3 that the individual nucleotides are connected by covalent bonds between the deoxyribose sugar and phosphate molecules. The alternating deoxyribose sugar and phosphate molecules form a backbone to which the nitrogen-containing bases attach. Okay, I'm sorry, I'm a little sketchy on this because I'm having trouble with it myself. Okay. So it looks like deoxyribose and the phosphates basically are the structure of DNA. Note also that the nitrogen-containing bases face towards the center of the helix and that they are perpendicular to the sugar-phosphate backbone. By facing towards the center, the bases on one chain of DNA face the bases of the other chain of DNA, with which they form bonds called hydrogen bonds. So the two chains are connected by hydrogen bonds. The locations of the hydrogen bonds are indicated in figure 10.3 by the dotted lines. Again, it's just this picture. I don't understand how it indicates it, but someone out there probably does. Alright. The hydrogen bonds help hold the two chains together. Hydrogen bonds are different from... Oh crap, I just lost my... Okay. The covalent bonds and ionic bonds that you read about in chapter 2. A hydrogen bond is relatively weak, I already knew that, bond that usually forms between molecules. Hydrogen bonds form when two atoms share a hydrogen nucleus, one protein. The hydrogen bond that forms between the bases in DNA form between a hydrogen atom and either an oxygen or nitrogen atom. Notice that the base pairs are of uniform length because in each case, one base is a double ring purine. So purine have two, and pyrimidines have one ring, right? Okay. The form of DNA that is most commonly found in living organisms has a right-hand twist, with each full turn consisting of 10 base pairs. So it's like a ladder that's twisted right, and it has 10 base pairs. Complementary base pairs. As figure 10.3 shows, cytosine pairs with guanine and adenine pairs with thymine. The DNA nucleotides normally pair with these combinations. These pairs of bases are called complementary base pairs. Two rules, called base pairing rules, describe the pairing behavior of the bases. These rules simply state that the cytosine bonds with guanine and adenine binds with thymine. So this part's pretty important. Cytosine with guanine, adenine with thymine. I always just thought in my head CG, AT. So at least I knew AT, I thought of at, and what was left was CG. So I always used that. Complementary base pairs are connected to each other by hydrogen bonds. We already went over that. Note that cytosine and guanine form 3 hydrogen bonds, and adenine and thymine form 2 hydrogen bonds. As figure 10.3 shows, the nucleotide sequence in one nucleotide chain of the DNA molecule is an exact complement of the nucleotide sequence in the other chain. So one chain connects to like the helix splits in half, and one half is over here and one half is over here. So it's like the rungs are split down the middle, and the two sides are complementary to each other. So like you'll have like A over here, or adenine over here, and thymine over here. So they're complementary, they go together, and then like cytosine would be over here and guanine would be over here. So when they split apart, they all match up to each other, and they're basically a template for to replicate. I mean, we haven't gotten into that yet, so I'm not going to go too far ahead. Okay, we still have, I don't know if I can still upload this, but this might be part 1 because we're nearing on 11 minutes, so I hope this helps. It's kind of helping me. Thanks.